Assalamualaikum warahmatullahi wabarakatuh. So, today my group will present about CRISPR Cas9 in livestock. So, cluster regularly interspecies palindromic repeats, or we call as CRISPR Cas9, is a gene editing technology causing a major upheaval in biomedical research. So, CRISPR Cas9 is a unique technology that enables geneticists and medical researchers to edit parts of the genome by removing, adding, or altering section of the DNA sequence. So, by using genetic engineering methods, it's possible to obtain animals with new qualities like resistance to various diseases, increased productivity, or change characteristics of milk, meat, and blood. So, by using CRISPR-Cas9 technology, many rabbits have been created in the world. So, this is because the rabbit is the smallest animal used both in laboratory practice and in animal breeding, from which recombinant pharmaceutical proteins can be obtained on an experimental and industrial rabbits. Rabbits also quickly reach sexual maturity, have a short gestation period, and can produce numerous offspring.
So, in order to conduct this research, firstly, the genomic DNA of the Californian rabbit breed was extracted from the tip of the ear. Vector NPI program is used to perform the selection of primers, construction of recombinant DNA, and also restriction analysis. Other than that, the PCR procedure was performed on a DNA amplifier. Next is the extraction of plasmid DNA. The extraction of plasmid DNA was performed using the GeneJet plasmid mini prep kit with some modifications by the classic method of alkaline lysis and phenol chloroform extraction method. Alkaline lysis is the method of choice for isolating circular plasmid DNA. It is probably one of the most generally useful techniques because it is fast, reliable, and also relatively clean way to obtain DNA from the cell. While phenolchloroform extraction of DNA is a commonly used method for removing proteins from nucleic acid. Then, the quality and quantity of DNA of isolated plasmid and restriction fragments were then evaluated visually in UV light after electrophoresis in agarose gel. DNA from agarose gel was isolated using the GeneJet gel extraction kit. The size of DNA fragments in agarose gel was evaluated with DNA ladder mix. And to create the CRISPR-Cas9 components, a PX330 uses chimeric BBCBH HSP Cas9 plasmid at gene plasmid 42230 PX330 was used.
Injection of plasmid into the zygote. In this research, they are using female California breed rabbit at the weight of 2.5 until 3.5 kg and at the age of 1.5 until 2.5 years old. First, they will induce superovulation at this rabbit. This causes them to produce more zygote. Next, this rabbit will be introduced to chorionic hormone. After 17 until 19 hours of introduction of this hormone to the rabbits, the zygote will form. The zygote form then will be removed surgically. Next, the zygote will be washed in manipulating medium. Then, the zygote will be transferred to the freshly prepared manipulating medium under the mineral oil. Lastly, the zygote will be injected with the plasmid at the male pronucleus of the zygote. Cultivation of zygote to blastocyst. The cultivation of zygote to blastocyst will be done in vitro in an optimal condition, causing the blastocyst to be formed. The blastocyst then will be collected and observed under fluorescent microscope using blue light. This is to observe the condition of the blastocyst. Next, the blastocyst and the embryos that are in late stage development of cell will be collected and stored at -20 degrees Celsius for further usage.
Now, let's take a look at what are the applications used in genetically modified rabbit using CRISPR or Cas9 technology. Genetically modified rabbit can be used in production of pharmacological protein with milk. Firstly, it can produce drugs to treat patients with hereditary disease. So, how will it be produced? The promoter regions of the gene of tissue-specific protein of cow, goat, or sheep milk contain casein and whey protein. This promoter regions is used for producing transgenic animals. Transgenic animals are the producer of biologically active proteins with milk as part of genetic constructs. Those transgenic animals are able to synthesize human protein exclusively in the mammary glands and secrete it into milk from which it can be extracted. Promoter sequence of the whey acidic protein WAP gene in rabbit milk were used as part of recombinant DNA in the production of first transgenic animals. In 2014, the US Food and Drugs Administration approved the first drug to be recombinant human C1 esterase inhibitor is extracted from the milk of transgenic rabbits. This was designed to treat acute angioedema in patients with hereditary disease. Secondly, milk from genetically modified rabbit also can be used in production of rotavirus. What is rotavirus? Rotavirus is the virus that can cause severe watery diarrhea, vomiting, and fever. Children who get rotavirus disease can become dehydrated, as the rotavirus is the leading cause of viral gastroenteritis in newborns, so the component protein VP2 and VP6 of virion can be considered as potential candidates for vaccine creation. So, how was the rotavirus vaccine produced? Transgenic rabbits that secrete recombinant proteins VP2 and VP6 in milk were obtained, whose milk could be used as a source of rotavirus antigen. GM rabbits with the VP6 gene integrated into the beta-casein locus were obtained using the Cas9 technology to create a rotavirus vaccine. For this purpose, there were used micro-injections containing Cas9 in the form of mRNA and a donor DNA vector with the VP6 gene into the cytoplasms of rabbit zygotes. So, a transgenic rabbit with the correct integration of the VP6 gene can produce an experimental milk-based rotavirus vaccine.
Next, the application of use in GM rabbit using Cas9 technology is it produced GM rabbit's model of such human disease as cardiovascular. Based on the article Genetically Modified Rabbit for Cardiovascular Research by Jian Ling Fan said that transgenic rabbits generate by pro-nuclear microinjection investigate many gene functions associated with human disease. Transgenic rabbits has been used in human disease because in comparison with mice and rats, the rabbit heart is similar to that of humans in both structure and function. For example, human heart in which beta-myosin heavy chain accounts for 90% of total myofibrillar myosin. Rabbit heart is composed of 80% beta-myosin heavy chain, which is different from the mouse heart predominated by 95% alpha-myosin heavy chain. GM rabbits also used in immune disease including immunodeficiency, infectious disease, a metabolic syndrome due to both nutrition and heredity, Wilson disease, a copper metabolism disorder leading to severe hereditary disease of the CNS and internal organs, and other use in disease resistance.
So the benefit of CRISPR-Cas9 in genetically modified rabbit is more cost effective, which is it can reduce the costs that have been used as compared to other biological or chemical production technique. Next, by using this CRISPR-Cas9 technique, the green fluorescent proteins expression in developing embryo in vitro would be an effective marker of successful site-specific integration of a single copy of the transgene. So why is green fluorescent protein is a good marker? So green fluorescent protein is a good marker because of its useful properties such as high stability, minimal toxicity, non-invasive detection, and the ability that can generate green light without any external cofactor and without need of application expensive equipment. So therefore, the other benefits of this CRISPR-Cas9 technology
it will help to obtain the transgenic rabbit producer of biologically active proteins with higher efficiency. Last but not least, the benefit of CRISPR-Cas9 in genetically modified rabbit is it can create genetically modified that produce recombinant proteins with milk instead of whey acidic protein.
So, in a conclusion, livestock animals are important for agriculture economy and biomedical research. This is because they are sources of milk, meat, organic manure, and other products. The developments of genome editing technology, especially in CRISPR-Cas9, have revolutionized the generation of the gene-edited farms animals. So, for your information, the developments of CRISPR-Cas9 for genome editing has led to a range of novel ideas addressing challenges associated with modern livestock, including productive and fitness traits, animal health and welfare, environmental preservation, and impacts of human health. So, this technology allows molding the animal kingdoms and the environment as never achieved before in order to pursue human purpose to enhance global well-being. So in the end, probably sooner rather than later, this technology will be applied as part of the globalization we all live in.